moving. I'm not going anywhere.
Italian.
Yeah, yeah.
This is great time to travel, right? The airline prices are so low. So
Yeah, it's very cheap,
travel.
but you cannot fly, but it's very cheap
Oh.
to buy the tickets.
It's so funny. So people are arriving, but I think we can start since I have a few minutes to introduce you, and so maybe other people will join us very soon. So first of all, I want to thank all of you to be here. This is the fifth appointment. I really thanks a lot to the two speakers. We have today outstanding speakers today. And of course, you can find on the web page of this series, you can find the announcement of the next seminars that will be 24 November. And we will have Michelle Battle-Fisher from the Wright State University, USA, and Ranjao Ji from the University of Milan. So some very brief technical information before we can start with the talks. Each seminar will be about 40-45 minutes, and we will have for each seminar a discussion at the end. If you have a question, and of course are super welcome, please write your question on the chat, and I will read the question for you. We are recording the seminars since the speakers acknowledged to record them, and we are now on live on YouTube channel. Our first seminar will be held by David Kessler. He received the BS from MIT, and he got PhD from Princeton. He's actually professor at Bar-Ilan University in Israel, and he works between nonlinear dynamics, statistics, mechanics, and system biologists. He's a fellow of the APS. David, please, the stage is yours. We can hear you. You have muted. Good. You are muted. Still. It's on the left. There is a microphone. It's on the left. On the other side. Okay, you are still muted. But I can help. Maybe I can do for you.
Okay, here.
Okay, good.
All right, now let's share screen again. Okay, now you can hear me?
Yes, perfectly. Yeah.
Oh, okay.
Yeah.
Okay. Full screen mode again, and let's try again. Okay. Okay, anyway, but yes. So first I wanted to thank the organizers for inviting me, and hopefully we're done with the technical problems. So the title of the talk is Biological Networks Regulating Sulfate Choice Are Minimally Frustrated. This is work that I did with Herbie Levine at Northeastern, and his student Shubham Tripathi, who's officially a student at Rice, but when Herbie moved to Northeastern, he sort of moved along, so he's somewhere delocalized between Northeastern. And the work that I'm going to talk about appeared in PRL this year, and so if you want to read more, you're invited to look there. All right. So first, the motivation. Where did this topic come from? And the people to blame are Francesco, Stefano, and Caterina. There was a workshop at the Weizmann Institute where Stefano talked and about this paper that appeared in 2018 in PNAS called Topography of Epithelial Mesenchymal Plasticity. And what the paper did in short is they looked at a Boolean model of the EMT, the epithelial mesenchymal transition, and they looked at the properties of this model. They simulated the model and reported on their results and drew all kinds of conclusions. But when I listened to the talk, in fact, I was just shocked. I was just totally, I didn't know what to think. Why was I shocked? First of all, before we get there, what's a Boolean network? So the idea is instead of looking at differential equations, things become simpler for physicists, at least, to think about when every protein or perhaps microRNA in the network is a spin, and which is up if it's expressed and down, highly expressed, and down if it's not. And then if protein J regulates protein I, then the coupling Jij is 1, and if it downregulates it, it's -1, and if they doesn't affect it, then Jij is 0. And then you have an update rule, which is in fact just the 0 temperature update rule for a spin system. In other words, if the total effect of your neighbors says point up, then at the next time you point up. If the neighbors are telling you to point down, then you point down. And if they can't make up their mind, then you do nothing. And the update is asynchronous. You just go pick a random node, updated spin, pick a different random node, updated spin, and continue doing this until you get to presumably some stable, steady state where nothing at that point happens further. All right, so what did they find? The thing that really set me for a loop is they reported finding 1,198,287 steady states. That is, to say that's unexpected is like being kind. Simple models, like the kinds of models that Herbie had looked at, the ODE models for the system, give something on the order of 3 states, depending on the details of the model. You typically have an epithelial state, you have a mesenchymal state, and the whole point of Herbie's game was that there could exist perhaps some 1 or 2 hybrid states in the middle, depending on parameters and so forth and so on. And his claim was, in fact, that these hybrid states have some important biological role, but that's neither here nor there for the purposes of the thing. The point is that instead of getting 3 steady states, 4 steady states, 1 million is a little bit much. And if it's true, then the basic paradigm, the question which has been, the idea that's been floating around for the longest time is that a steady state of what is a cell type, right? Because we know that although all the cells have the same programming in principle, they all have the same genetic instruction set, but nevertheless, there are the cells adapt different phenotypes. And the idea was that there's some dynamical system describing the regulatory network of the cell, and the cell can fit to one of a number of steady states, and those describe the different phenotypes. So, and then the question is how many cell types there are in the human body. It's a number of order. I don't remember the number, but it's something on the order of 40 or something like that, slightly less than 1 million. So how are we supposed to understand the million steady states? And furthermore, how could a cell function with 1 million different steady states? Because that means that anytime you perturb the cell, you're not going to go back to where you came from, because you're going to fall into some other basin of attraction, because basically the entire phase space of the cell is partitioned into so many different little packets that you do anything to the cell, it's going to knock it to a different steady state. And then what do you do then? So, and worse than that is typically when you have a system with that many steady states, your natural inclination is to interpret this the way a physicist interpret this, that what we're looking at is nothing but a spin glass. Because we know that a spin glass has exponentially many steady states. And so the typical, if I see a spin system with many steady states, I'd say, and that's what I said, this should therefore be a spin glass. And now a spin glass is, why is it, it's a glass. In other words, the dynamics is frozen. You get stuck in one of huge numbers of these metastable states, and you never find the point, you never find the lowest energy state. You never get even close to that because you get just stuck. And so the dynamics is totally frozen out. That's not what you expect for to think of a cell dynamics as being a spin glass is also unacceptable. So now this is not a problem that is unique to the problem of regulatory networks of cells. In fact, I worked in problems in ecology and trying to answer what is essentially addressed exactly the same issue in the ecological framework. In ecology, if you try to write down a model for interacting species, right, some kind of generalized optical para where you have competition between species, then you encounter what's known as the biodiversity problem, which was discovered by Robert May a long time ago, and who proved that, in fact, it's very difficult to create a dynamical system which has a stable, steady state with many different competing species. So it's easy if you have 2 species, 3 species, 4 species, no problem to find a model with a steady state. He was looking for a single steady state, but in fact, and systems with small numbers of steady states are of measure 0 in the space of all dynamical systems. So what you typically get is some kind of spin glass, and again, you have millions, huge numbers of steady states. But you don't get what you expect in ecology, which is maybe 1 operating point or perhaps 2 operating points of the ecological system, not millions of operating points. All right. So where does that leave us? So the question that immediately rises to the surface is the gene regulatory network a spin glass? It looks like a spin glass. It smelled like a spin glass. Is it a spin glass? Now, a spin glass is just a random assortment of ferromagnetic and anti-ferromagnetic interactions. Well, that's what we have. And so it seems very reasonable that if you try to put together some kind of, if you have some matrix Jij, and it's got both ferromagnetic plus interactions and minus interactions, why shouldn't you get a spin glass? But the question is then, a spin glass is when the Jijs are assigned randomly. That's when you get a spin glass. So is, in fact, the gene regulatory network essentially random? So let's test the idea. That's what we decided to do. So the idea, what we did is we took a network, an existing network that's supposed to describe, in fact, we started, in fact, with the very network that Stefano and company looked at, this model of the EMT. That's how we started, and we decided to randomly rewire the network. So we go in, and the idea is to screw it up and see if anything changes. Because if it's a random network, then randomizing random gives you the same thing. So if you see no change in the system upon randomizing it, then in fact, the system is acting as if it was a random network. So what we do is we rewire the network. If protein A induces or inhibits B and protein C induces or inhibits D, then we switch the link. So now A drives D and B drives C, and otherwise, so we go in and 1 by 1, we rewire the network. And the nice feature of this kind of randomization is preserves the topological, quote unquote, features of the network, the things that network science looks at, which are in and out degrees of nodes, the number of plus minus links, all the various, macroscopic features of the network are unchanged, and yet it's a different network. And then the idea is to compare the ensemble of rewired networks with the original, quote unquote, biological one, the one that was supposed to describe the EMT system. All right, so in fact, in the end of the day, we wound up looking at 5 different models. One of which is the model of the 72 node model for the EMT transition. There's a simpler version of that model, a 26 node version of that model, There's another model which has to do with stem cell transitions from stem cell behavior to differentiated cell behavior that has a model with 52 nodes and 123 edges. There's another model for small cell lung cancer, and then there was another model we found for gastric cancer. So these models we didn't make up, we just took them from the literature, so we're not responsible. Presumably, you know, we're just going to take them, the people who constructed these models, at their word. And when we looked at the models, what did we find? That, for example, we know that there's a huge number of steady states in the 72 node EMT model, because we were told there were 1 million. We weren't so ambitious. We stopped after we found 300,000. We found 30,000, and that was good enough for us. But if you keep going, you'll find more. The stem cell kind of potency model had a 4 × 10⁵ steady states we found. The gastric cancer network is a smaller network. It only had 1,200 things. The 26 node network had even smaller, 104. The small cell lung cancer was sort of exceptional. It had more nodes. It had 33 nodes and 357 edges, but it only had 10 steady states. And that's an interesting thing. And I think that going to lead into what Mohit is going to talk about in the second talk today. But anyway, these were the 5 models that we looked at. And now here are the results. So this is the key slide here, because this is what we found when we did this. See the 5 models presented. And now I have to explain to you what we're looking at. What we're looking at is, in fact, so what we did is we looked at either the entire collection of steady states that we found. That's what's shown in the green. The green is the steady states we found, the entire collection of steady states we found for the randomized networks. The orange is not the entire, is weighted, is the trajectory, the endpoint of trajectories. We ran each network, we ran for some number, for 500 different initial conditions or whatever. And then we ran it for I forget how many, 50,000 time steps or whatever it was. And wherever the network wound up at the end of that time, that counts as 1 point. The blue and the pink are the same thing for the original biological network. So if we take a look at the 72, we'll start with B, which is the 72 node EMT network. All right. You notice that if you look at the randomized networks, and what are we measuring? We're measuring the frustration of either the steady states or the endpoints, whether we're talking about the green or the orange, of the state. So what is the frustration? We look at every bond in that state and we see if it's in fact doing what the Hamiltonian is telling it to do. So if we have the other spins that are talking to that bond, are telling it to be up and it's down, then it's a frustrated bond. If it's pointing in the correct direction, quote unquote, then it's not frustrated. All right, so typically what we find is that, roughly speaking, for the random networks, 1/4 of all the bonds are frustrated. And that's when we look at all the steady states. If we look for where the states that are involved that are actually the basins of attractions, it's even slightly more frustrated. And you get the distribution of frustration is basically some Gaussian centered around 1/4.3. If you look at the biological network, the original Frank Close, et cetera, Stefano Caterina model, again, this typical steady state also has high friction. But you notice that there are states with very, very low frustration. And in fact, if you compare the blue to the pink, the pink, you see, goes down as far as the blue, but in fact, there are many more endpoints with low frustration. In other words, that the states with low frustration have a high basin of attraction, much larger basin of attraction than the states with high frustration. So it's not, so both the random network and the biological network have high frustration states. So if you look at the entire collection of steady states, they don't look all that different. It's only by noticing that there are states with extremely low frustration that you see a difference. And then then we looked at these other networks, and lo and behold, they share the same property, that in all these 5 models, there are essentially no low frustration states in the random network. There are, however, low frustration states in the biological network, and they have a very significant fraction of the basin of attraction is in fact these low frustration states. And that works for all 5 networks. So we have managed to discover some difference between the biological network, the original biological network, and its randomized version. So the implication is, in fact, that the biological network is not random. It's not just a spin glass. It's not just any old idiot wired the system at random. In fact, there's some special design. It is designed and has special features. We don't understand yet, what the meaning of these special features are, but in fact, there's a clear sign that there are special features. So once we do this, we can actually ask other questions. We can look at the overlap distribution. So we look at the steady states and we say, do they resemble each other? So if you look at, and we did this for the EMT network and the stem cell network. So the top row is the EMT network. The second row is the stem network. And if you look at the randomized network and you look at the overlap distribution, you see it's centered around 0. It's a wide distribution centered around 0. Whereas if you look at the steady states, the low frustration steady states of the biological network, then in fact, it's very much a bimodal distribution, strongly peaked close to +1 and -1, very different than the behavior of the randomized network. Now, it's interesting that in the original PNAS, they also looked at the overlap distribution, but in fact, they had weighted them they looked at all the steady states and not just the low frustration steady states. And since there are so many states and they didn't weight them by their basin of attraction, so this feature didn't stand out in the original thing. It's only when you focus on the low frustration states do you see this clear signature of the bimodal overlap distribution, which if you look at all the steady states and weight them, just give the same weight to every steady state independent of its basin of attraction, then that wipes this feature out. You also see a similar difference in the phenotypic score. By looking at what proteins are expressed, we can come up with some measure of epitheliality, which is blue if it's mesenchymal and dark red if it's epithelial. And if we look at the steady states of the biological network, we have these extreme values of most of the states are mostly epithelial and purely mesenchymal, purely with high frustration of the states in the center of this diagram of this. And in fact, they have neither epithelial nor mesenchymal tendencies, are sort of smack in the middle. And the same thing is true for the stem cell network. All right. Okay. So that was phase 1. So now, is it not biologically relevant? I mean, the cell is not operating at 0 temperature, noise-free environment. The cell is quite deep. So does this all go away suddenly if we turn on noise, or is this robust to the addition of noise? So we can introduce noise in one of 2 ways. One is by doing a thermal kind of noise, a pseudo temperature, so that the update rule is the update rule corresponding to a finite temperature, not zero temperature. And what happens when we do that is that the frustration goes up, and the minimum frustration also, the mean frustration goes up a lot. However, if you look at the low frustration states, they don't go up. They're still low frustration states, even in the presence of significant thermal noise, quote unquote, pseudo-thermal noise. Another way to add noise to the system is to do not just a total rewiring job, but just look at the effect of a few miswirings. And again, the low frustration states still persist. There's still this tail of relatively low frustration states, even in the presence of significant, we changed 60 edges and still there are these low frustration states that are still present. And as we increase, if we look at the stemness score, which is like the epithelial score, it's very bimodal at low temperatures. It gets less so, but still it has a definite bimodal characteristic at the highest temperatures. And same thing for what happens with the degree of rewiring. All right. So now the question is, how does nature produce low frustration networks? Because we don't have the idea why the network to us looks random, and you don't see anything, just like studying the JIJs, at least offhand. So how would you produce a low frustration network? So if in the absence of any other knowledge, the question is, can evolution do it for you? So what we did is we created a population of 500 different networks. Every network is mutated with some small probability. That's the mutation rate. Mutation being a single link switch. Each network is evolved for 5,000 time steps for each of 50 random initial conditions, and note the lowest frustration endpoint. And that's basically the fitness of the network, is how low a frustration state can it achieve. And then what we do is, out of these 500 networks, we choose the 25 with the lowest frustration endpoints and use them to populate the next generation. And then we monitor the statistics over generation. And the results are like this. Looking at the major graph, the main panel, what we see is the population average of the minimum frustration endpoints of the network versus generation. So this is, low numbers or high fitness, and this is the fitness over time. And in fact, the systems become very much more fit. They started out with a random value, which was a frustration of 0.3. And at the end, they wind up with numbers around 0.025. In fact, some of them get to lower frustration endpoints than the original biological network. And if you, and this is the inset is the average of the frustration of all the endpoints over all the networks versus generation. And there's a similar trend that the evolution helps. So in fact, if nature somehow was selecting because if minimum frustration states are what's necessary to work, then you can imagine that they are have higher fitness, and the system would evolve in that direction. Similarly, you could look at in this evolution experiment, you can look at the evolution of the overlap distribution, and it goes from being this very wide distribution at generation 0. And as the generations increase, you see we're building up to this bimodal distribution characterizing a non-random, quote unquote, biological type network at the end. So we still don't, this still doesn't tell us what it is, how to look at a network and or how to construct a network ab initio that has this property, but the evolution algorithm is able to find it, to find networks like this. All right, so summing up where we are so far. So these biological Boolean spin networks exhibit a state of very low frustration steady states, a set of very low frustration steady states. These low frustration states have a much larger basin of attraction than the high frustration ones. So they're the ones that are more dynamically relevant. The set of the low frustration states has a very bimodal overlap distribution. Their randomized network counterpoints on the other hand, don't have any steady states with this low frustration. So the frustration is significant. This is a significant effect. I mean, the chance that you would by random chance have generated a biologic, these low values of frustration are 10 to the minus 6, 10 to the minus 7. They're impossible, or even, no, they're even worse than that. It's 10 to the minus 8. I forget the numbers, really low numbers. The low frustration states are relatively robust to noise, and a network with low frustration states can be evolved from a randomized network. So that was our first pass at this, and then we gave this to the referee, and the referees, we had one particular referee who asked us all kinds of questions, which were instructive. So the major question that concerned him was a question that we didn't even think of, which was he wanted to know, maybe in fact our biological models are not representative of reality, because these biological models were created by people, and they were good models in the sense that they did what they reproduced the known phenomenology of the networks, right? If they hadn't done that, then these models would have been considered a failure. So maybe in fact these models are biased. They're not have nothing to do with nature. They have to do with the way people construct the models, and they basically stop when they put in just enough complexity to do what it wants, but they don't add additional complexity, which would screw everything up and make it spin glassy. So what he wanted to know, which is a hard question, was would a model derived directly from the biological data without human intervention and / bias show the same effect? So we tried to make him happy. What we did is we used expression data for normal breast cells to construct a regularized regulatory network governing this MYC phenotype. We started with 398 genes known to be associated with the pathway. We obtained this transcription factors for the genes. We used this program GRN Boost 2 to obtain the association between the genes and the transcription factors, remove the genes that didn't regulate any other genes, and that produced a network which we didn't put in by hand. It came to us out of this GRN Boost. And so this is a data-driven model as opposed to a human, curated model, if you will. And the question is, does this data-driven model have low frustration steady states compared to its randomization? So this is the answer. The two things on the left are the randomized versions of the network we constructed. The two things on the right are the data from the model itself. So this model that we didn't construct, that the computer constructed for us, shows low frustration states. This by itself didn't make the referee happy, because he was afraid that somehow the way we were randomizing was too extreme. That what he wanted, he thought maybe in fact there's still a bias, even though it was now not human bias. Maybe the GRN Boost algorithm, not that I have any understanding of how that works, but maybe that is generating the bias. So what he wanted us to do was shuffle the inputs to the program and see if the program would produce states with low frustration. And the answer is they have lower frustration than the truly randomized things, so much higher frustration than the quote unquote biological model using the real expression data. And therefore, indeed, even taking a model given to us straight out of the computer, the effect persists, which is very comforting to know. Another issue that he addressed was, what about more general Boolean models? Because this simultaneous, this additive model where the energy is Jij, Sij, is one kind of a Boolean model, but in fact, there are much more general Boolean models. And Stuart Kaufman, in his work back in the, I guess, the 70s and 80s, considered model networks with more general Boolean functions. And does this make a difference? So in order to deal with that, we needed a more general definition of frustration, which is to look at the effect of one protein on a second protein, and we trace out all the other inputs that sort of determine the size, the sign of the spin, as it were. And then we studied a model of, in fact, gonadal sex determination, which had these kinds of, which was this kind of a network. The model had 19 nodes containing groups of ands and ors and not gates, giving 79 edges. So it compounds and A plus B, A and B, or C, not D, something like that. It controls the output of F, whatever. And indeed, again, the model has low frustration, whereas the randomized versions have much higher frustration. So even if you don't take the simple Jij, Si, Sj models, the same kind of phenomenology exists. All right, so what about, the last thing is, what about ODE models? Because there's the same thing apply. If we leave the world of Boolean models behind, the ODE models work the same way. So we use this recipe algorithm, which generates couplings for a given network architecture. And we use this to generate couplings for the 26 node, the smaller EMT model. We looked at 500 different sets of parameters, which were spit out by the algorithm. And we ran those 500 different models with 50 random initial conditions for each. And we did this for the original EMT wiring diagram, and then we did it for the randomized wiring diagrams. And what we did, what we did at the first pass is we looked at, instead of looking at overlaps, because the variables are concentrations, they have all kinds of different scales. Each protein has a very different, vastly different levels, so you have to put them all on the same scale. So to make life easier, we looked at pairwise correlations of particular nodes, and we see that, interestingly enough, there's a difference again between the random networks and the original network, the EMT network itself, that the couplings were either, pairwise correlations were either plus ferromagnetic, anti-ferromagnetic, or nothing, or non-interacting, whereas in the random networks, the correlations were all over the, centered around 0 and peaked around 0. And in fact, when we looked at the evolving, we had this evolving network architecture that we had looked at with the evolution experiment. So now we look at the evolving network architecture, look at it versus the recipe ODE models of that versus the models of randomized versions of that, looked at different generations. And again, we started out with a model which was characteristic of a random network, and we finally get at the end to a model which is characteristic of the biological model. You can, in fact, define, if you work a little harder, define an overlap distribution. What you have to do is, if you shift, you look at the log protein levels, shift by their average amounts, and then normalize by the standard deviations, then you get a spin type of variable, which you can then use to compute an overlap between two different configurations. And again, what we see is the random networks are centered around 0, whereas the EMT network has this bimodal distribution. Here, it's not quite as bimodal as it was before, because this is looking at all the steady states, not the low frustration steady states, because we didn't have a good definition yet of frustration. So that's something we still need to work on. All right, so I have a few minutes left. And this is where where our thoughts are running to now. So up to what I said up to now is what's in the PRL, and we've trying to think of where we're to take this next. And one idea that we came up with is, what is the relation of these ideas to this notion of sloppiness in system biology models of Sethna and coworkers? So Sethna invented this concept of sloppiness, saying that most parameters in biological models are not very important. And there are only a few directions in parameter space which are relevant, but most things, in fact, most parameters are not relevant. So that if you're trying, he was thinking about trying to fit a model to experiment, and when you're doing the fit, it's sort of undetermined because most parameters the system just doesn't care about, and there are only a few parameters which are important. That gave rise to this notion of sloppiness. So there's something like that going on in our game. So that's the claim from Sethna, is that most models in systems biology are sloppy, and in very few directions which model parameters can be varied to change the model behavior substantially. And so then he has a quantitative measure of that, which is calculating some error function, how things change when you change parameters, looking at some determinant of some second variation of that, and looking at the eigenfunctions of this Hessian. And his definition of sloppiness is when you have very stiff modes versus the majority of modes being very non-stiff, very sloppy, relative to the stiffest modes in the system, and that's the sloppiness. And so we did that for our models, and for the 26-node EMT model, the dark green is random networks, the sloppiness of the random networks. The purple is the biological, and in fact, what you see, this is the this is the average of the different parameter vectors, the ratio of the lambdas. So small is sloppy, and higher values are not sloppy. And in fact, the biological model, the original EMT model, is less sloppy, it would appear, than the randomized model. Yes, than the randomized model. So here's the sloppiness. If you look at it again, the minimally frustrated states are somewhere in between the random networks, which are much more sloppy, and just random sensitivity matrices. Just to give you a scale, if you pick random Hessians at random, they are much less sloppy. So the EMT network has this immediate sloppiness, but it goes in the wrong direction because the model, the biological model, is less sloppy, it turns out, than the randomized versions of the model. And that's an interesting observation, but we don't have any idea yet what to do with that observation. So to sum up, what we have is that, these biological, remind you, these biological networks exhibit a set of very low frustration steady states, both for spin networks and the ODE networks. And the pure JIJ, SISJ, and more complicated Boolean models, they all have the same phenomenology. The low frustration states have much larger basins of attraction than the high frustration ones. A set of low frustration states have a very bimodal overlap distribution. The randomized network counterpoints don't have any steady states with this low frustration. The low frustration states are relatively robust to noise, and a network with low frustration states can be evolved from a randomized network. So the takeaway messages are one definite one is that, so in one way of thinking about this, biological networks, the network problem, design problem for biological networks is sort of like the protein folding problem. The original question in protein folding was, how does a random protein fold? And it took a long time to realize that, in fact, don't fold. So if you're asking about the dynamics of biological networks, the answer is, how do they function? Well, random networks don't function at all. They're spin glasses. They're not going to work. And biological networks have to be designed, and now remains to be seen. The other question, the conclusion is more of a speculation. And the conclusion is, how are we supposed to now think of a cell phenotype? It clearly, if what we're seeing is at all indicative of reality, then you can't think of the cell phenotype as a single steady state, and you have to, at the very best, think of it as a cloud of steady states with similar features. And then how to talk about such a cloud of steady states. This is not like a quasi-species, which is a single steady state of an evolutionary process with perturbed by noise, the noisy cloud around a single final steady state. This is in fact a cloud of steady states, which is a different concept. Now, maybe in the presence of noise, the distinction becomes not relevant, and it's just a cloud as a cloud. But anyway, that's where we are at the moment. So thank you for your attention.
Thank you so much. Very, very interesting. I'm sorry that Jim Setna is not here to discuss with us, because me and Stefan and Gina Setna discussed a lot about these sloppy models, EMT, when we were in Cornell, and very, very interesting, this last part for me, and you got me some ideas. So please, questions?
I have one.
Please.
I wonder if you try to reduce the number of nodes and evolve from random network to get a low frustration states. What happens if you reduce the number of nodes? Is there a threshold where you find qualitative uniform results, or not? Or you can reduce it until you find a trivial threshold?
We haven't tried it at all. No. Yeah, so it's an excellent question, and we haven't tried it. We haven't tried it at all. The thought didn't occur to us, so we'll have to go back and look at it.
Thank you.
Yeah, I also have a question. Hi, David. So in the beginning, you said that you were shocked by our results of having 1 million steady states, right? Which seems like crazy, and I was very, very happy to see that.
Right.
And I wonder, after all this fantastic work that you presented today, which I found very interesting, and especially the randomization bit. So I wonder, what is your view now, right? Are you still shocked by having so many steady states, or a little bit less, maybe?
Well, I mean, it's still a challenge. It becomes less of a challenge.
Okay.
Because if the dynamically relevant steady states are these low frustration states, and they're all, so that reduces the magnitude of the problem from millions to dozens or 100. And if all those final steady states, these low frustration states, all have high overlap with each other, then in some sense they're all the same, perhaps functionally the same. And so, and then the system doesn't care which one of them is, because they're doing the same thing. Perhaps. I mean, that's what my idea is, that this, a phenotype is this cloud of steady states, so that it doesn't have to be one. If, as long as all of them are, we have the group, the ones that are all the same, when you have this broad, you had a broad Q alpha overlap distribution, so all of them are very different, then it's impossible. But if we're restricting ourselves to the low frustration ones, which seem to be the ones that are more dynamically relevant, and especially once you add in noise, then they're the ones that hopefully the system, falls into that area of phase space, whatever. And then the problem is, I can imagine that might be okay. But I mean, that remains to be seen. I mean, it reduces it from an impossibility to something that is still a challenge, but something that, I can imagine works out in the end. But that's all I can say.
I see. Okay.
Yeah, I have the same one. Yeah, there is a question of Tony Giorgino. I don't know if he wants to talk directly, or I can read.
No, yes, I can.
Please.
First of all, thank you for the amazing talk. And if I understand, you are implying or suggesting that resistance to noise may be a reason selecting the low frustration networks. And if it is so, do you think that you could adapt your genetic algorithm to show this, so that the genetic algorithm goes, instead of going towards low frustration, goes towards noise resistant networks, so that you could demonstrate this?
That's a good question. You should try that, yes. I think that probably it will be true, but we haven't checked it. So, but I think it's quite likely. I mean, if they're, to the extent that these seem to be correlated, it hopefully will, right? Because to measure frustration, right, I mean, of the steady states is something that, you know, is a little unnatural for the biological system to do. I mean, so, you know, looking at robustness to noise is probably a better way, more realistic way of trying to implement it. But we have it actually, so we should try. Another idea, another homework assignment.
Next paper.
Yeah.
Good. Okay, other questions? So very interesting. And actually, since we have the YouTube, we can send the video to Jim, because I think it will be very interesting to watch your presentation, your talk. Yeah. Thank you so much. Really, thanks a lot. Thanks a lot. So we can move to the next speaker of today. Thanks a lot, David. So we have Mohit, Kumar, and Jolly. So today we have two friends. Mohit leads the Cancer System Biology Group at the Center for Biosystem Science and Engineering at the Indian Institute of Science. He's also fellow of Indian National Young, Young Academy of Science. He's also secretary of the International Epithelial Mesenchymal Transition Association and co-chairs of the Mathematical Oncology subgroup at the Society for Mathematical Biology. So he's very active, he's really working a lot. Thank you to be here, please.
Can you see my screen, Katrina? Great. Thank you so much, Katrina and Stefano, for the kind invitation to present some of the work that we have been doing. And it's great to have David in the same session, because some of the ideas that he introduced will directly be relevant as I talk about some of the themes, both the ideas as well as some of the tools that he mentioned, receipt, randomization, and ODE models is something that you will hear about more. So my focus is, or our focus is particularly in looking at different regulatory networks implicated in cancer metastasis. So if you look at the process of cancer, where it initiates in one or more cells and then forms this primary tumor, this palpable mass, which is visible to the clinician, and then it no longer remains restricted to that particular organ, rather spreads to different organs in the body, which is the process called metastasis. This is the typical journey of most solid tumors. The time scale can be different for different tumors. What I'm trying to highlight here is the acceleration that happens in the process once cancer begins to spread. Now, over the past couple of decades, we have made some fantastic discoveries in identifying cancer early, in cancer prevention, in identifying the molecular mechanisms, what genes are involved, what lifestyle factors are involved, such as the association of smoking with lung cancer. But most of these fantastic developments restrict themselves to the initial stages of the disease progression. And there has been really no major breakthroughs when it comes to restricting metastasis. And that's what we focus on. It still remains the cause of 90% of all cancer-related deaths, and cancer cells spread from one organ to another, largely through the circulatory system in the body. Now, when I started working on this as a graduate student with Herbie Levine at Rice University, what I found most intriguing was that if you think of the most common cancers, they do not have the intrinsic property to start moving and start invading. Because more than 80% of cancers begin in epithelial organs, such as breast cancer, prostate cancer, lung cancer, liver cancer, etc. So they are stuck in a specific 3D geometry, have a specific topology, topography there, and a specific function to perform that comes from the structure of the organ. So how do they gain this ability to move? And once they have reached a distant organ, they again lose this ability to move. Where is this switchability in both directions coming from? And metastasis is not just about this switchability, it's also about other axes on which cancer cells switch their behavior in this reversible spatiotemporal manner, which is their ability to adhere to their neighbors. They have to be able to evade the attacks which are being made by immune system. They have to be able to settle down on a new organ, which is completely different when it comes to the biochemistry and or biophysics, biophysical properties. So we look at metastasis as a dynamical process, and it has been shown that metastasis is actually a highly inefficient process. The success rate is less than 0.02%. But then what is so special about that small percentage of cells that are finally able to metastasize is what we try to understand. Now, when we think of cancer, one idea that popped up is the idea of mutations. And mutations have been associated with various different stages of cancer progression, but so far there is no unique mutational signature that can be pinned onto the process of metastasis. So this makes a lot of sense to me, because in the process of metastasis, when the objective of cancer cells is continuously changing, having an irreversible fixed solution, hardwired solution, such as a mutation, which has very little chance to evolve, it only propagates from generation to generation, that is probably not the best strategy. And what cells might be adapting is non-genetic or a non-mutational mechanism through which they are able to alter these different traits simultaneously without necessarily gaining new mutations. And this adaptation need not necessarily happen at a single cell level. There may be some division of labor in a colony which is migrating together, and clearly it has been shown that when cells move together, they form much more metastasis as compared to when they move as single cells. So in contrast to the genetic paradigm, which is heritable, irreversible, deterministic, what I'm trying to talk about is the non-genetic paradigm, which is not necessarily heritable. There is an element of heritability which may be built in based on the molecular mechanisms involved. More importantly, it's reversible, and there is an element of stochasticity involved. Now, the idea that whether cancer can proceed without mutations, let me, to emphasize on that, let me take a digression and talk to you a little bit about bacterial populations. So what has been seen over the years is that if you take a bacterial population and then treat that with increasing dose or time of antibiotic, what happens is that a large number of cells die, but there is a small number of cells which is left behind. Now, if you take the small number of cells, so this from this biphasic curve, this population, and then regrow it, what you see is that this graph is reproduced across generations. Now, if the resistance gained, or resistance that these population of cells is showing, was due to genetic mutations, then what you would have seen is more of a flat line, because you're not changing the antibiotic, you're still using the same antibiotic. And if they have adapted to it once irreversibly, then you would have seen basically no effect of that antibiotic on that population, because that population is going to be genetically identical. However, that was not seen, and over years people have realized that this is not happening at a genetic level, but actually at a phenotypic level. So without change in mutation, without change in the genome, there is a change in the phenotype that is happening, and the ideas of multi-stability have been established there. Similar ideas of reversible drug resistance are beginning to come up in cancer, and they have also come up in viral populations and so on. So if there are such ideas of non-genetic heterogeneity in drug resistance, can similar ideas also be applicable in the context of metastasis? These ideas are again not new to cancer. These ideas do form the basic tenet of embryonic development, where if you do have this progenitor cell which has the capability to give rise to any of those two cells, now which cell does it give rise to? There is this element of stochasticity that is involved here, and which is also being trying to be harnessed for the process of cellular reprogramming and so on. So these ideas of non-genetic heterogeneity, phenotypic heterogeneity, plasticity, switchability are becoming more common day by day in the cancer literature. And especially with the advent of high throughput technologies where you can actually do sequencing at a single cell level, one can then do various different nonlinear reductions of data collected, as shown in one example here, and identify what different cell types are present. But what I'm going to talk about today are sort of more fundamental questions that one can ask. Where is this heterogeneity coming from in the first place? What is the role of this heterogeneity in the context of cancer metastasis? And eventually, can we control this heterogeneity in a dynamic evolving system? So I moved to India 2 years ago to start my own lab and the Cancer Systems Biology Laboratory, where the overall goal is to develop mechanism-based mathematical models to look at both metastasis and therapy resistance. And we work together with various different clinicians and experimental cancer biologists to develop these models. So the three basic tenets of the work that we do, which I'll show you a glimpse of today. One focuses around the molecular basis of phenotypic plasticity. What networks are involved that allow these cells to switch? What is the name of those different molecules, and how do they connect? What are the design principles of those networks? Why are networks designed the way they are? And this sort of directly follows from what in the previous talk David was mentioning. And then eventually trying to connect it to clinical applications. Can we have biomarkers as well as some new therapeutic ideas that can be explored there? So I have different lab members working on each of these different things, and I'll show you a glimpse of them. We have looked at some of this work in the past 2 years, and there are 3 recent lab members who are still undifferentiated, who are just joined and still in the Waddington landscape of projects trying to decide what exactly to work on. So since David has already introduced the idea of EMT, I'll quickly go through it. The idea being that in the solid tumors, which are predominantly epithelial, do not have the property to migrate or invade. The dogma in the field had been that they undergo this transition called as EMT, where they now do gain the ability to migrate and invade, and usually form the first step of metastatic cascade. And once they reach a distant organ, they undergo the exact opposite transition, where they no longer need to migrate or invade, but actually settle down and form a whole new tumor. So this EMT, MET paradigm for the decade or so, in the previous decade, had been largely seen as a very binary process, where all these cartoons that are taken from leading reviews in the field of EMT and metastasis do show that cells are basically, at the end of the day, moving as individual cells, and whatever intermediate states they may have are referred to as metastable, and the possibility of cells moving collectively was not really mentioned. Either a cell does not move at all and stay epithelial, or it moves as single cell. What has happened in the past couple of years is that there have been both mathematical models, including the one from Stefan and Katrina and Frank, and the one we did earlier on a much smaller network than what they worked on, showing that it is possible to have a third stable state, which we can call it a hybrid epithelial mesenchymal state, which can have the property of collective migration because it has still not lost all cell-cell adhesion from epithelial, and it has begun to gain some properties associated with mesenchymal, such as migration or invasion. And this idea has been seen in multiple different cancers and is becoming a leading idea in the field of EMT in general. It has also been proposed. We initially had a systems biology-based model for this, and there have been enough experimental follow-up now saying that not only these cells exist, but actually they are potentially fittest for the process of metastasis. If you can deplete them, you can actually reduce metastatic burden significantly. And this is from a Nature paper that appeared 2 years ago, where they identified multiple different states of EMT and showed that the ones that were most metastatic were actually the ones in between, not the ones which were completely epithelial and not the ones which were completely mesenchymal either. So when I started the group here, we wanted to first look at some gene expression data, some clinical data, or experimental data, and see can we quantify EMT in that high dimensional landscape a little bit better than what we had been doing earlier. So we had developed a model earlier, a statistical model to quantify the EMT-ness of a sample on a scale of 0 to 2, where 0 would mean fully epithelial, 2 would mean fully mesenchymal, and if the score is right around 1, that would mean maximum probability of it being hybrid. There were 2 other metrics which were there in the field. So we first checked that whether these metrics were performing well enough, and they actually showed very good correlations with one another, showing that despite having different genes and different algorithms that they were using, there was some global EMT signature they were capturing. Then we looked at all these different cancer cell lines belonging to different tissues. And for each tissue type, we calculated the mean and variance of different EMT scores using the different cell lines. And what we observed was that, again, the ones that those were heterogeneous were the same, irrespective of which method you used. And interestingly, those were the ones which were enriched in having hybrid phenotype, suggesting that the more the hybrid phenotype, the possibility, the more the variability that you would see in the EMT-ness per se. And there is a reason for that, because hybrid cells tend to usually lie in between epithelial and mesenchymal, and as I'll talk about, relatively less stable. So it is possible for them to switch. It's easier for them to switch to other states, and potentially that is why they give rise to this high variability. The other disease we looked at particularly was called inflammatory breast cancer, or IBC, which is a highly aggressive disease. And there's still no molecular therapy out there. What was there in the literature is 4 different gene lists that people had identified using different algorithms. And the first question we asked was that, is there any common gene in these gene lists? And there was none, with only one exception where you look at the 132 gene list and the 50 gene list. There's no other common gene, which was very, very surprising to us that, if people are giving out these gene lists based on different clinical samples, one would expect some common molecular processes or genes to show up, but that they did not. Then we again used the 3 EMT scoring methods, and we had previous experimental data to suggest that this might also be a more hybrid EM phenotype like cancer. And what we did observe was that if there was one common feature connecting these 3 different data sets across these 3 different EMT aspects, that was not the mean of EMT scores, that was actually the variance in EMT scores. So suggesting that this particular tumor is, there's no unique gene list for it, but if there is one unique feature that we can say so far, it's actually the higher variability, which again, as I talked about, can potentially be mapped onto the hybrid phenotype in general. This is the network that we had looked at a while ago. We started with this very simple network, and there there are these 2 transcription factors driving EMT. There are these 2 microRNAs that drive MET. So if the levels of these molecules is high, the cell is epithelial. If the level of these molecules is high, the cell is mesenchymal. We had a framework to capture microRNA, mRNA regulations, and so on. Long story short, we made 3 different predictions 7 years ago, saying that first of all, EMT is not binary. You can have cells which are in this hybrid state, as shown by this branch in the bifurcation diagram, where x-axis is some EMT-inducing signal, and the y-axis is one of those proteins which depicts the degree of mesenchymalness. The other, since you can clearly see the regions of multi-stability, what we predicted was that if there are cells with the exact same genetic background, you can still see them in different EMT states. And true to multi-stable region, if there is enough noise, you can also see a so-called spontaneous switching without necessarily having an induction per se. All these predictions were validated both by our collaborators and others independently over the past couple of years. These are non-small cell lung cancer cells stained for an epithelial and a mesenchymal protein in green and red respectively. And we cultured these cells over multiple passages over 2 months to see whether it was not just a static snapshot, but actually a phenotype that can be propagated and maintained in the lab. So you clearly see that most cells here do have both red and green proteins, suggesting that this is a hybrid phenotype and not just a random mixture of epithelial and mesenchymal cells. This feature, using the same two proteins that I talked about here, vimentin and ecadherin, we can see that this heterogeneity does exist in multiple different cell lines. This is no exception. You see this in breast cancer, prostate cancer, lung cancer, and colorectal cancer, that there is this heterogeneity despite having the same genetic background. There was this experiment which was done where they actually sorted these 3 different cells, which were either ecadherin positive, vimentin negative, vimentin positive, ecadherin negative, or double positive. So double positive would be the ones that are hybrid. These are the ones that are epithelial in green, and the ones in the violet color would be mesenchymal. So they were segregated and cultured independently. What you saw was that over a period of 2 weeks, they switch. Epithelial cells and mesenchymal cells largely maintain their phenotype, and the hybrid ones are the ones that switch the fastest. We have developed detailed models for that, why this particular phenomenon is seen, but I don't have the time to go into that right now. I'll be happy to talk about it later. So what we wanted to next understand was, are there factors which can stabilize this hybrid state? So, in presence of these factors, is there a larger region of parameters in which this hybrid state can exist? So this is the same network that I talked to you about, and we found this interesting protein which was actually doing very counterintuitive things. On one hand, it was promoting EMT, on the other hand, it was promoting MET. So we were confused as to what exactly this is going on. So this is the same bifurcation diagram that I showed you earlier on a different scale. And when we plotted the bifurcation diagram of this entire circuit, we saw that there was a larger region of parameters in which this hybrid state existed. You plot the dynamics and you see that in presence of this molecule, the progression to a complete EMT is actually slower. It takes much more time, therefore suggesting that in presence of this molecule, you can actually see hybrid state for a longer period of time. So the prediction was that, okay, let's go back to the cells that maintain a stable phenotype and delete this protein and see what happens. So these are the same cells that I showed you. If you delete this particular protein, you see that the red molecule disappears, and these cells have become basically completely mesenchymal. And that is also shown in their migration and invasion behavior, not only just at the molecular level. So both at molecular and functional level, we were able to see changes which are consistent with a complete EMT, suggesting that this particular molecule was acting as a brake on EMT. As soon as you remove that brake, cells basically undergo a complete transition. Now, since we started looking at EMT networks, there have been various other groups that have come up with different EMT models. They've used different modeling strategies, both at ODEs and also Boolean levels. So the questions we wanted to ask is, are there signatures in these network topology that allow for hybrid states? Because each of them is a different model, uses different formalism, so it becomes hard to compare the predictions being made. So this work was led by my PhD student Kessler, some students who helped him, Buran and Vishnu, in collaboration with Frank, Stefano, and Katrina. And again, the initial discussions of this were held at the same workshop that David referred to, the Weizmann workshop in 2018, where we started discussing whether some of these approaches were possible. So what we did was we used different networks and simulated them in 2 different ways. You have heard about both of them from David. One is Boolean, so the parameter independent approach, where we follow the Ising model kind of formalism and look at the steady states obtained depending on the initial conditions. And other where you do use an ODE approach, but you randomize all different parameters, and for every parameter set you start with different initial conditions. So you get steady state space, but you also can get the phase space there. So this is a parameter independent and a parameter agnostic approach, and we compare different networks of different sizes with it. So what did we find? This is just a description of receipt, where you basically enter the topology file. A inhibits B, B inhibits A, which gets converted to an ODE, and all these parameters are sampled. And this is the Boolean approach that you've already heard about, the sigma, Pij, Sj, which is what is used to update the value of a given node when you update nodes in an asynchronous manner. We use this metric called Jensen-Shannon divergence to quantify the difference in the steady-state distributions obtained. The lower the value of JSD, which varies from 0 to 1, the closer the distributions are. So we looked at all these different networks, GRHL2, NRF2, OC4, and so on, and simulated using both receipt and Boolean. What you see is that the most common steady states can be seen irrespective of what simulation strategy you follow, and also the value of JSD is relatively low overall. Suggesting that the steady state distributions largely depend on network topology, not necessarily the specific kinetic parameters that we are talking about. Then we did all different kinds of perturbations. We deleted an edge, we changed the sign of an edge from negative to positive and positive to negative, and we added hypothetical edges. And then we calculated the JSD of every such mutated network with every other mutated network. So here's that matrix. So what we see is that there is one network that stands out when you do Boolean approaches, and there are two networks that stands out when you do receipt approaches, and one of them is common actually. So suggesting that again, both receipt and Boolean analysis can identify which perturbation will take your steady state distribution far away from that of the wild type or the control case. And we also did this more further quantification where in a given plot, every dot is a perturbed network, and we calculated the JSD from wild type based on Boolean, based on receipt. Again, we saw a very good positive correlation overall. Now, what we were interested in is saying that, sure, you have multiple states and these distributions, but that doesn't tell you whether those states coexist or not for a given parameter range, for a given parameter set. So we defined this, what we call plasticity score in two different ways. One was just the frequency of parameter sets that would give you more than one state. So that's and you can calculate the fraction of that. And other is a more biologically motivated definition where you take all the solutions and then project them on this Z and mid 200 axis. Again, the double positive is hybrid and the single positives are epithelial and mesenchymal respectively. And we ignore the double negative population here. And then you see in how many populations, in how many parameter sets you actually see these different states. So this is basically like trying to identify, if you are plotting bifurcation diagram, what percentage of that bifurcation diagram can correspond to some sort of multistability. So again, we saw that these scores correlate irrespective of the network that you look at. The red one is the wild type case, the control case, the biological network, and the blue one is every single perturbation that we just talked about. Interestingly, what we saw was that some perturbations increased the plasticity score and some of them decreased it, saying that if you make certain perturbations, you are making the system more likely to show multistability, and others you decrease it. So we wanted to see if this can be predicted from JSD, and unfortunately it could not. So if you correlate JSD with a fold change in the plasticity value, there was no correlation whatsoever. So then we started asking, well, what enables multistability in the network? And there have been some suggestions that positive feedback loops, at least when looked individually, can enable multistability. So what we did is in every network, we counted how many positive cycles are there. So you basically start from a node and trace all different paths that come back to it and keep multiplying the sign of those edges that form that path. And if the overall multiplication sign is positive, it's a positive feedback loop, but if it's negative, it's a negative feedback loop. So again, what we see is that we both did the kind of perturbations that I just talked about earlier and these randomizations where you swap the edges. So you don't change the in degree or out degree of any node. You don't change the total number of negative or positive links in a network. You do possibly change the in degree of negative edges or inhibitory edges and in degree of activatory edges and so on. So again, what we saw was, again, irrespective of which network you look at, not only biological networks, but also random networks, but also randomized networks, the higher the number of positive feedback loops, the higher the chance of plasticity, suggesting that positive feedback loops are contributing to multistability. And if you can break them, you can restrict the ability of cells to switch back and forth and potentially therefore restrict metastasis as well. This experiment was done. And they actually looked at the same feedback loop, which we had started modeling initially. So using CRISPR-Cas9, they actually broke this feedback loop And in this mutant case, what they observed was that the metastatic count, which is shown on the y-axis, was actually much smaller as compared to the wild type case, where you have this feedback loop, which is very intact. Please bear in mind the y-axis is on log scale, so the effects that you see here are quite both statistically significant and biologically significant. Now, this paper doesn't really go into reasons of why this happens, but we think and potentially map it onto what I talked about, that if you break feedback loops, you reduce the cells, you reduce the ability of cells to switch, you reduce heterogeneity. So there may be some cooperation that is going on, some division of labor that is going on among those cells, for which there is some experimental evidence. And you basically deprive the population of both switchability, adaptability, as well as the chances of cooperation. Simultaneously, you can do both things. So summary of this part, the metastasis is a dynamic process. The hybrid cells are perhaps the most fit, and positive feedback loops can block both EMT and MET simultaneously. Currently, there is no clinical trial ongoing or no inhibitors being developed which can do both things simultaneously. You are either trying to block EMT or trying to block MET. If you're trying to block EMT, you may be helping cells which are already in the metastatic cascade, because if you block EMT, you may be helping them in settling a different organ and so on. And this approach was actually recently highlighted in a Nature Reviews Cancer, saying that perhaps we should start rethinking about how we are trying to block metastasis. And one of the ways we should think about is, fix the position of the cell on the axis, not let them move left or right. They are fixed where they are, deprive them of their adaptability feature. Switch on to the other aspect I want to talk to you about, which is different from EMT, but you'll start to see similarities in a minute. Small cell lung cancer. So this is contributes to 10 to 15% of all lung cancers, causes 250,000 deaths per year worldwide. That's exceptionally aggressive. If you compare the 5-year survival rate to breast cancer or other cancers, this is extremely small, less than 7%. There's no new treatment strategy in over 50 years, no molecular targeted therapy, and it relapses very quickly after initial response. It's not a new cancer per se. We have known this for almost 100 years now. There were what the community calls a forgotten disease age, in which there was a lot of TCGA and other analysis done for various other cancers. Somehow SCLC was excluded out of it. We have information about the cell of origin. What we know is that across all different patients, there are these 2 mutations which are seen. We have cell lines, we have mouse models, and all different high-dimensional data that exists for this. Again, this is a typical case where mutations don't explain everything, because the P53 and RB1 mutation is seen in every single SCLC patient. So where is this heterogeneity that I'll talk about coming from in a minute? It's clearly not due to mutations, because mutational background is identical as far as these 2 genes, the most important ones that are mutated, are concerned. So the ideas of multiple states and the possibility to switch among them in the SCLC literature has been around for 30 years. It has not been very well studied relatively. So there are these 2 phenotypes which people typically call a classic phenotype and a variant phenotype, which you can clearly see there is a difference in how they grow in suspension. And there were recent experiments suggesting that if you have both of them, that actually leads to a much more aggressive disease. So potentially there's some cooperation that is going on in this context. Again, like EMT, the initially SCLC was characterized into what is called neuroendocrine and non-neuroendocrine in this or classic and variant in some manner. But clearly we have realized over time that this is not a binary categorization. There are cells which can be in this so-called hybrid neuroendocrine, non-neuroendocrine feature. So this work was led by 2 undergraduate students, one majoring in biology, Lakshya, and one majoring in physics, Uday. So they came together to work on this, and also supported by my PhD student Kishore, who led the previous work that I talked to you about. The questions we wanted to understand was, again, is there multistability in the network that underlies SCLC? What networks are multistable, and are there features of network topology? This was the network that was published a couple of years ago. As you can see, it's a much larger network than what I've talked about. This is a 33 node network with 357 edges. So we were expecting that this would give many steady states, just as you heard David talking about 1 million steady states in the 72 node network for EMT. To our surprise, we found only 4 states. And those 4 states, basically, we did Boolean simulations here using the same Ising model approach there. And we started with 2 to the power 20 initial conditions, 2 to the power 25 initial conditions, the same thing. We used RASCP, which is the continuous version of this, 10 to the power 6 parameter sets, 1000 initial conditions per parameter set. Again, largely we saw these 4 states. We did see some other states, but their frequency was less than 1% or less than 3% or so on. So again, suggesting that there is something fundamental about the topology of this network that allows so few number of steady states, so few phenotypes, despite such complexity. So then we did the experiment of randomizing these networks, where again, in randomization, you basically swap the edges among themselves. And for each of those 1000 random networks, we did Boolean simulations and saw how many states did those 1000 random networks give rise to. And this is the distribution. And the x-axis, please bear in mind, is on the log scale. So every single random network that we tested gave much more number of steady states than this wild type network or this biological network. And none of these random networks gave rise to any of those 4 states. So if you look at JSD, the JSD value was basically very close to 1. So then we asked another question. We said, okay, now let's do perturbation. Let's delete an edge, or let's change the sign of an edge and see what happens in that particular case. So for every single edge, we calculated JSD for these 2 perturbations, deleting the edge and changing the sign of an edge. Out of 357 cross 2 perturbations, 678 cases had JSD very close to 0, saying that if you do those, 340 plus some perturbations, they make no change whatsoever to the output of this network. If you delete that edge, or if you reverse the sign of an edge from negative to positive or positive to negative, it doesn't change. There was only a very small percentage of cases which were actually giving JSD, which was significant in any manner. So this suggested to us that possibly there's a lot of redundancy which is built into the network, that if you delete a single edge, it just doesn't crash. If you completely shuffle everything, then of course the system crashes. But if you delete a single edge, there is enough redundancy, enough robustness that is built into the network to maintain the phenotype. So then what we did was we did these pairwise correlations both for Boolean and receipt simulations from the steady states that we obtained. So here, these are the 33 elements that you see here. And what we saw was very interesting, that looked like that there were these 2 teams of players which were positively correlated with one another, but within a team. But when you looked at across teams, they were negatively correlated. So this suggested to us that it is possible that this is a toggle switch, but instead of having just one gene on both ends, there are 2 teams of players who are inhibiting each other and self-activating themselves. So then we asked this question, is this feature specific to this topology? So what we did was we basically quantified this metric here. So these values will be positive, these values will be negative. So you can define this particular metric here. And you can calculate the same metric for 1000 random networks. And again, this is the distribution of the 1000 random networks for this metric. And this value, 496, is the value for the Boolean simulation biological network. Again, saying that biological network, this particular feature of 2 teams is specific only and only to the biological network and is not shown by any other random network. This trend, surprisingly, was also seen in the experimental data. You use the same 33 genes and you see this kind of trend that shows up here. But now, this is only correlation. It just doesn't tell us how these genes are regulating each other. So we asked this question, if 2 genes, X and Y, are negatively correlated, it can happen due to multiple reasons. A, X is inhibiting Y, directly or indirectly. B, Y is inhibiting X. Both are inhibiting each other, or there is no inhibition among, there is no connection among themselves, but they have a common upstream regulator which is activating one and inhibiting the other, and so on. So we define what we call influence matrix, where for every pair of nodes, we define this metric where we looked at, can you quantify, if you sort of go from node I to node J and take all different paths less than, say, path length 10, and then quantify what is the sign of each of those paths, and then weight them based on how long the path is. The longer the path, the less the weightage, because the direct regulations are likely to be more important than the indirect regulations. What we saw was that this influence matrix was basically very similar to the correlation matrix. So then we again calculated this correlation between the correlation coefficients obtained from simulation data and influence coefficient. Now bear in mind, this influence matrix does not need any simulations. All it needs is topology. Based on topology, you can actually come up with this. And we saw that there were very good correlation. Now, suggesting that yes, there is a reason why these correlations are seen, because the network is designed in that manner that there is mutual activation within a team, and then there is largely speaking mutual inhibition between the two teams. So we again wanted to test this. We took all the different 1,000 random networks, and for each of these random networks, we had these influence correlations, and we regressed them against the receipt correlations and tried to identify what the value of correlation coefficient was. So in this particular case, in the biological network, this value was around 0.8 or something. In random networks, most of these values were very close to 0. The peak of these distributions is at 0, no matter which data you take, simulation data or the experimental data, and so on. So then what we did was we actually took that large network, and based on the connections, based on this influence matrix, we were able to reduce it to a much smaller network. And this network is sufficient to explain all the features seen. You see the 4 states, those 4 states are basically the same, when you project them from 33 dimension to this 4 dimension. And again, whether you use Boolean or you use recipe, you get the same results. So 2 of these molecules, one of which belongs to this group A and the other one, that is neurod 1 here, are actually known in the literature to be able to categorize SCLC cell lines and samples and so on. So we wanted to see, are they really master regulators per se? So this is just the simulation data where we did observe an overall negative correlation, which is what is known in the literature, that the ones which are ASCL1 positive are typically neurod1 negative and vice versa. So we took 50 different cell lines and then did hierarchical clustering, and we saw these 4 clusters very clearly. Now, when we project them on ASCL1 and neurod1, beautiful segregation. These 4 clusters are basically the 4 quadrants. So you can have cells which are double positive, double negative, and single positive. And we did other means of clustering to show that 4 was actually the best number of clusters that perform, optimal number of clusters, and we saw the same clusters. Interestingly, if you look at our simulation data that I showed you earlier, these are the exact 4 states: A positive, N negative, A positive, N positive, A negative, N positive, and A negative, N negative. So based on that network, we are actually able to explain and say that, yes, these 2 molecules do explain this heterogeneity quite well. So here what we saw was that there was not one molecule which was one set of molecules inhibiting each other, but there were 2 teams which were inhibiting each other, and they are constraining the network solution space in such a manner that despite this complexity of 33 nodes and 350 links, we basically see only 4 states, each of which has a biological meaning in this context here. Now, this teams of toggle switches is not something which is unique to SCLC. We have also seen this in another context. So we were looking at a fatty liver disease context. And again, we saw that there were 2 teams which try to maintain the liver cell state. There were 2 teams which are trying to, molecules forming a team which are trying to take the liver cell state and try to push it to a fat cell state or an adipocyte cell state. Again, when we simulate this, you see these 4 clusters show up very clearly: high, low, low, high, double negative, as well as double positive. Another instance where we saw this idea of teams of toggle switches is in the context of when cancer cells try to adapt when they're deprived of matrix. So when they're deprived of matrix, they typically die, but some of them are able to switch to another state reversibly, that they survive. If you look at the network again here, you see the same phenomena. There are these two teams of players inhibiting each other. Here, this is not transcription factor. This is a set of enzymatic reactions, phosphorylation and dephosphorylation, but the concepts hold the same. Here, we were fortunate that our experimental collaborators here did this experiment, which was done in EMT earlier, that if you have multistability, you should see switchability in absence of any inducer. And this is what was seen. When they segregated these two different phenotypes here, you were able to see that they were able to give rise to the other phenotype without necessarily having any other induction. So that's all about toggle switch or teams of toggle switches. What if you have a more complicated structure, such as a toggle triad? A toggle triad means that A, B, and C all inhibit each other. So these are like three toggle switches which are connected in a triangle. So we wanted to see what exactly can be the output of the system in this case. So what we found was something very interesting, because this network is actually intrinsically frustrated. Because if A inhibits both B and C, and then B and C also inhibit each other, it's difficult for the network to arrive at a steady state. So what we saw was that this network gave rise to 6 states. 3 were more dominant as compared to the others, and those 3 were single positive. So either A was high, so the capital letter indicates the state, the node that was high. So either C was high, B and others were low, B was high and A was high, and these were comparable. Or these double positive, where both B and C were high, A and C were high, or A and B were high. This was again done using receipt. And the triple positive and the triple negative were extremely small. Interestingly, we found that this topology is actually seen in T-cell decision making. And you see all these phenotypes, at least 5 of these phenotypes have been reported experimentally as stable phenotypes. So of course, there are these 3 differentiated phenotypes, Th1, Th2, Th17. Also, there are these hybrid phenotypes, Th1, Th2, and Th2, Th17. These have been reported experimentally. One of them, we made this prediction that this can be a stable phenotype. This work was led by my PhD student, Achita, who's a fantastic artist also. So he gave this rendition of a Waddington landscape in this context, saying that this network, this is how this network behaves. You can have these 3 states, and you can have these 3 hybrid states present in between as well. So I'd like to just summarize that cell-fate decisions, be it in a disease context or in a developmental context, and I presented various different examples of it, do involve multistability. And typically this happens through either a toggle switch or a couple toggle switches in different forms, or teams of toggle switches. And decoding these underlying organizing principles is potentially crucial for understanding these decision-making aspects, as well as designing circuits. Now, I'd just like to end with a note here, and that is, just a comment on how experimental biologists versus theoretical physicists looking at this approach can have different meanings of the word understand itself. Because when I present this to a bunch of biologists, many of them are like, well, we already knew what the circuit was. Sure, you can know what the circuit looks like, but unless you have information about the time scale, length scale, strength of regulation, combinatorial effects, etc., you can come up with all different predictions about the outcome of this model. And that again goes back to the sloppiness idea that David was talking about. And why this becomes particularly important in the context of cancer is because there have been cases where the therapies that were being tried, or at least in clinical trials, such as the anti-angiogenesis therapy in the early 2000s, saying that if you block the blood vessel growth, you have taken care of the primary tumor growth. And that therapy had exactly opposite results. When you block angiogenesis, what was observed was the metastasis was accelerated, because cancer cells are clearly switching to a different state when you're trying to push them in a corner of a room, saying that you can't do this anymore else. So we need to understand this from a process as to how these different axes of plasticity are regulated, are connected with each other, and how are those decisions made. So I would like to end with this call for all the physics audience here, saying that sure, there are these different networks, but as biologists also realize, and these are the last sentences of the most cited reviews in the field of cancer biology, which the first one written 20 years ago saying that cancer biology still doesn't have that logical coherence that rivals that of chemistry or physics. And 10 years later, when they rewrote, they still said that, you know, there are no underlying organizing principles. We'll see what they write in 2021 or 2022, but my guess is it will not be very different from it. So with that, I would like to thank the fantastic set of people I've had the fortune to work with and collaborate with in this iterative theoretical experimental aspect, because clearly cancer is a complex system, adapts in all different ways, both at cellular levels and at tissue levels and so on. And we do need inputs coming from different disciplines to try to really understand that from a quantitative, predictive aspect, and eventually being able to make some intelligent therapeutic interventions based on that. I would like to thank you for your attention, and will be very happy to take any questions and discuss further.
Thanks a lot, Mohit, for this very nice talk. I take over from Caterina on the sharing of the session. So are there questions that somebody wants to ask? So I would say you have found some sort of organizing principles, no? That there is a common theme, this multi-stability, and can you comment more on this issue of we've seen that in large networks you have a lot of states, and in other networks that are also large you have a discrete number of states. So, and can you comment on what would determine this? So what are the organizing principles that would give you small number of states or large number of states? And can you also comment on which kind of systems you expect? Because you have many pathways, no, that describe different features. Right. And we have seen some pathways like EMT is very multi-stable with many states, and others that are less so. So can you comment on this dichotomy, on whether you can switch from one type of the other, whether is really a dichotomy or there is some intermediate thing in the middle?
So I think that is what we are actually trying to see, if there are other large networks which have so less number of states. Because most other large networks, including the one that you guys looked at, give rise to many, many more states. Now, clearly some states are relatively less stable. So one can say that, yes, the epithelial and mesenchymal pool of states, or the cloud of states, as David was saying, are perhaps more stable as compared to others. But we are yet to come across any other large network. And when I say large, I mean more than 25 nodes or 100 edges overall, which has such few number of states. Once we look at that, then we can probably comment more on the organizing principles aspect. From the point of view of cancer, I think that in certain scenarios, I mean, the degrees of freedom are different, are likely to be different if you talk of different axes of plasticity. So with, say, metabolic reprogramming, people are finding that, again, it's not a binary system. You can have states in between. So we can continue to find states, but how are these states connected to each other? So we have seen scenarios where if you couple toggle switches, then you go from 2 × 2, 4 states to 2 states. So because some states sort of annihilate the stable and unstable state merge together and do that. So we are trying to understand those also as we build more and more networks across these different axes.
Yeah. So one comment, by looking at just by eye at the network you show that has 4 states, it had an impressive number of edges. So like everybody was connected with everybody. So while this is not true for the EMT, you have some hubs and so on. Did you try to characterize the distribution of the degree distribution to see whether this plays a role?
So we have not done that, but there was a recent paper in PLOS ONE, which I think looked at the network that you guys had studied, and then tried to identify what was called sort of periphery nodes versus hub nodes. And based on that, they were able to reduce the network. So we did that analysis. And in this particular SCLC network, there's no periphery node. So every single node has an input and an output. Or, I mean, at least one input and at least one output.
Yes. Thanks. So any other question? And post them on the chat, or even maybe talk, since we are just a few of us.
Yeah, I have some comments. So thanks for the talk, Mohit. It's very interesting. I was very surprised by the results. I was already surprised when
when I saw the preprint. So one comment is that, yeah, 300 edges for 33 nodes is a lot. It's very uncommon. And that, I think that is more of how the network was constructed by humans, more than something biological. I don't know. So, but that could be a difference in how the network was built.
Right.
And but still, so if I understand correctly your results, you say you find teams of players. So there's all these nodes in a team that go together, right?
Yes.
And so if they always move together, like they are 100% correlated, do they really represent different entities, or could we say that the effective size of the network is a bit smaller in a way? That
Yes.
would solve a little bit the...
Yes, yes.
But
So
it's just a possible view, of course.
Right. So we were able to reduce this to a 4-node network. And with that 4-node network, we were still able to see the same 4 states,
right,
which were there. So yes, it is possible to reduce them. Now that, of course, brings in, as you rightly pointed out, I mean, how much of this network is, truly existing versus our interpretation of the links, because this network was constructed by humans. So for now, we have taken this network at the face value and, not really gone into how that were there. But now, because in another context, we are still constructing that network. But when we do correlation based on experimental data, we again see the ideas that there are 2 teams possibly. So I think this may be a more general principle than this particular network. So sure, this network, may be even more very fine-tuned as compared to what it exists. I mean, there might be some refractive index there to reality. But I think the overall idea of having these toggle switches instead of single entities and entire teams is perhaps more generic.
I see. It's interesting because it can always happen that we get a network which is, I don't know, overdone. Yes. And so I'm interested now in this simplification of the network. It's a very useful tool, I guess. It's to know if a network is minimal, in describing phenomena.
Yes.
So it's very interesting. Yeah.
Yes.
Yeah.
So,
maybe we can talk another day. It's interesting.
Sure.
This tool.
I mean, currently we have done it in a relatively crude manner, but I'll be happy to discuss other
That's
ideas in graph theory or other aspects that you're aware of to do this in a more unbiased manner and at networks where there are more complicated and we don't know that there are only 4 states, for instance. But the idea of coming up with the influence matrix was that can we say something about the topology even without simulating the network? And there we saw that, yes, this influence matrix was basically a replica, more or less, of the correlation matrix which we obtained from simulations. So here we have a way, at least for these special networks, so-called, to give out information about them even without simulating them.
So I have now a question following up on what Franz says. So I wonder if this network was not created by just looking at cross correlation between genes. So I mean, this is a standard way to create networks. You just look at the transcriptome for many classes of genes and say, okay, all these genes are correlated. When it's correlated, I put a node. When it's anti-correlated, I put an inhibition node. If you do that, then of course, when you go back, you get that your influence matrix is correlated with the correlations, because that's how the network was constructed. So I wonder if you checked whether this network was constructed in this way.
So this network at least partly used this algorithm called weighted gene co-expression network analysis. So I think it is based on some of the ideas you are saying, but I think it goes beyond that to look for the not just correlations. So based on correlations, they might be identifying modules of genes, but what connections exist between them were identified using a more sophisticated approach.
Okay, so thank you. Are there other questions or comments? If not, I think we can thank both speakers for this nice afternoon and also for synchronizing their talk on the same topic, which was not intended, but turned out perfectly. And so I would like to give a virtual applause to everybody, and we'll see each other in 2 weeks for the next virtual seminar on complexity. Okay, thanks a lot.
Thank you so much, Stefano. Thank you.
Thank you, David. Thank you, Mohit.
Bye.
Bye-bye.
Bye.